Hello friends, welcome to my channel Bioimaging. Today we have a guest with us, Dr. Saumitra Chaudhary. Dr. Saumitra has completed his PhD from IISER Bhopal in the year 2016, where he studied the role of endocytosis in synapse development using the well-established Drosophila melanogaster neuromuscular junction model. During his postdoctoral research studies with Dr. Mahela Sarpe at the section on cellular communication, NICHD, he continued to pursue understanding of the molecular mechanisms governing synapse development and plasticity. He has received extensive training in genome editing, biochemistry, super resolution imaging, and single cell genomics working at the National Institutes of Health, USA. Presently, he is working as scientist 2 in the confocal facility CCRF, AIIMS, New Delhi. Today, his topic of presentation would be high resolution in vivo detection and reconstitution of synaptic proteins using a novel nanobody-based system. I welcome you, sir, and over to you for the presentation.
Thank you so much. It's a very kind introduction. I'm really touched by that. And thank you so much for also letting me present the work. And I think during these difficult times, I think this is a wonderful platform to reach out to people and reach out to younger students, I would say. And I hope like in the coming days, we'll have many other people also making use of this platform. So I'll start sharing my screen. Yeah, so what I'm going to talk to you today is going to be more about high resolution imaging. And since this is also more relevant to the channel, so I thought I'd be talking about high resolution in vivo detection and reconstitution of synaptic proteins using a novel nanobody based system. So this work was carried out during my postdoctoral training at the National Institutes of Health. And at the very outset, I'd like to just put on this disclaimer telling that this presentation is purely on my front and it is not endorsed by NIH or AIIMS for that matter. So just before getting started, since I think there will be a lot of younger students who are also going to see this presentation. So I would just very briefly talk to you about my career trajectory shortly. So I did my schooling from the northeastern part of India in Mizoram. And then after completing my schooling, I moved on to Bangalore, where I did my bachelor's and master's. And after that, in the year 2010, I moved into the Indian Institute of Science Education and Research for Bhopal for pursuing my graduate or PhD studies. And after completing that, I then moved to the United States at the National Institutes of Health for pursuing my postdoctoral studies. And earlier this year in February, I came back to India and I joined AIIMS as a scientist in the CCRF microscopy facility. So this was just to give you a very kind of a brief introduction to how my career has spanned. All right, so as Dr. Somnath has already mentioned, I've worked extensively on understanding synaptic development. So just to get started, what are synapses? So synapses are the sites of communication between neurons. So we've got like 100 billion neurons in the brain, and these are the connecting units. You usually have two neurons. One would be a presynaptic neuron, the other would be a postsynaptic one, and there is a passage of information from one neuron to the other. And if I have to explain this in a layman terms, I like using this paradigm, which is something that I am sure everyone of us would have actually either participated in or at least seen. So this is something called as the relay race, wherein you have it's a team effort. You have one person having a relay baton, and he passes on this baton to the other person, and this other person then has to run, and then he has to pass the baton to another person. So this kind of information exchange keeps happening in the brain, and just for the purpose of simplicity, I would like to tell you that this is a very fast information process or information exchange that takes place And this happens at the scale of microseconds. So you must understand that it's a very fast process, and it is also a very highly choreographed relay race. So going further, again, this is a cartoon representation. And how does this information exchange actually take place? Of course, you've got two constituents or two important ingredients, I would say. You have a pre-synapse and you have a post-synapse, and there are going to be multiple proteins on either side of the compartment. And this is a synaptic cleft, which is just like, say, 20 nanometers apart. And there are different proteins which are present on either side of these compartments. And there are some other proteins which even bridge the two compartments together across the synaptic cleft. So again, this is again a cartoon representation. So I've already shown you previously that you've got a presynaptic cell and a postsynaptic cell. You've got neurotransmitters which are packaged in this synaptic vesicles, and upon stimulation, they release these neurotransmitters into this 20 nanometer cleft. And then there are receptors on the postsynaptic side. These neurotransmitters then bind to these receptors, and then the signal is transmitted downstream. So I know this image looks pretty much overwhelming, but just to tell you the extent of the number of proteins that are present on either side of the compartment. So the postsynaptic side is pretty much rich in these receptors. You've got the kainate receptors, you've got the potassium channels, you've got the different glutamate receptor channels, the NMDA receptors, the kainate receptors, the inotropic receptors. And on the presynaptic side, you've got the entire machinery for vesicle fusion and endocytosis. You've got the neurexin neuroligin protein complex that bridges the synaptic cleft. You've got dynamine, CAST, and MINTs. These are the proteins that are involved in basically helping in the exocytosis of the vesicles. And then you've got a hell lot of machinery here, including dynamine and AP2 and clathrin proteins that are involved in the vesicle regeneration. So the reason I was showing you this is just to give you an appreciation of the number of proteins that you have on either side of these compartments. And again, as I've already mentioned to you, that this synaptic cleft is just 20 nanometers. The reason I'm stressing on this 20 nanometers, you will come to know across as I move further in my presentation. So I've talked to you about like how a synapse looks like, and that synapses are the fundamental units in the brain. Now, why are we really interested in understanding synapses and synapse development per se? Now, that is because any defect in synapse development can be linked to neurodevelopmental disorders. And these are terms that we've all heard at some point: autism, schizophrenia, learning disability, and there are many more. So there is a wide spectrum of disorders that can result when there is a problem or there is a defect in synapse development. And that's the reason why studying at the mechanistic level how synapses develop is very important. Now, the question is, how do you really study these defects or these disorders? Of course, you cannot get hold of a human being and then try to, study his brain just like that. So you need a model system. People use different models. It could be a cell culture model. It could be the invertebrate model like Drosophila, C. elegans, or it could be higher model organisms like mouse, rats, etc. So my studies involved using the modest developmental organism that's called Drosophila melanogaster. And the striking similarity of Drosophila with humans and the conservation of at least 70% of the disease-causing genes across both the species makes Drosophila a very wonderful model system to study synapse development. And what is shown in this cartoon is the corresponding organs. You've got the brain, you've got the ventral nerve cord, which is again analogous to the spinal cord. You've got the segmental nerves, which are again kind of the peripheral nerves in humans. So you have the entire system that you have in human beings, complex system, and you have a similar kind of system in fruit flies as well. And the other advantage, of course, is the life cycle of Drosophila is really it's really short. So you can start with an embryo and you can reach to an adult within 10 to 12 days at a temperature of 25. And you can also regulate this developmental cycle by rearing them at higher or lower temperatures. At lower temperatures, they'll grow a lot slower. At a higher temperature, they'll grow a lot faster. So as an average, anything between 12 to 14 days, you will have transition from an embryo to a fly. And over the years, we've also developed wonderful databases like Marvel, OMIM, and Undiagnosed Disease Network. Now, what these platforms provide you is a comparison of different model organisms, the disease genes that have been studied and associated, the relevant disease models, the relevant publications, and you also have a list of different mutations that are associated with the diseases. So it's kind of very elegant to look at these websites, look at these databases, get the relevant information, and then of course carry forward your work on a particular gene in a particular model. And of course there's a lot of clinical data available as well, so you can get in touch with the doctors and clinicians and of course collaborate with them and do a lot of work. So that makes it more relevant. So now, why study Drosophila, and why do we have to really consider the complex human brain, and why do we actually compare it with a fly brain? So what I've kind of put across here is the human brain, the mouse brain, and the fly brain. And if you, I want you to really look at the scale out here. You've got 100 billion neurons in the in human brain. You've got a scale of centimeters here. If you look at a mouse brain, which is relatively smaller, you've got 75 million neurons, and the scale is in millimeters. Whereas if you look at a fly, you've got 100,000 neurons, and the scale is in micrometers. So, you've gone relatively lower when it comes to the scaling. But then compared to the 100 billion neurons, the 100,000 neurons in flies also achieves a similar level of complexity. And that's the reason why it makes a wonderful model system to study neurodevelopmental disorders and synapse development per se. So, and again, the advantage of using Drosophila neurogenetics, you have different techniques now available, like you can do genome editing using CRISPR, you can do a lot of structural biology work, you can do a lot of high resolution imaging. Now, this is something that I'm going to show you quite a lot in the coming slides. And of course, you can do functional analysis like electrophysiology, protein biochemistry, behavior studies, and electron microscopy as well. So one model organism, and you can do, you can study a lot in this system. So now what I'm going to introduce to you is the Drosophila neuromuscular junction. That's our model system. And you're going to come across this particular structure in the coming slides. But then just to show you what exactly is the structure that you're looking at. So this is a third instar Drosophila larva. This is the ventral midline. When you dissect a larvae open, what you see is something like this. In green are the muscles. So this is a muscle surface. This particular organism is expressing GFP, and that's the reason why you see the green. And the central nervous system, which is analogous to our human brain, is labeled here with epitope called as HRP, horseradish peroxidase. And this is the ventral nerve cord, which is analogous to our spinal cord. And then you've got these fine thread-like structures. So these are the projections coming out from the ventral nerve cord. These are the axons. So these project out from the brain, and what they do is they innervate the muscles. So what you see here is these blue structures, and they innervate the muscles. And when they innervate the muscles, the way they look like are these rounded structures. And so this is the entire axon in blue. And then you've got these rounded structures which are innervating the muscles. Now, if you go further, zoom into one of these round structures, what you see is, so these are like 30 to 40 individual synapses. In red, you see a staining for a auxiliary subunit protein called netto, and in green is something called Brook pillet, which is an active zone component protein. Now, if you remember from the previous slides, I had talked to you about a presynaptic compartment, a postsynaptic compartment. So the presynaptic compartment here is represented in the green, that's BRP, and the postsynaptic compartment is here shown here in the red, and HRP, of course, is the neuronal surface marked in blue. And the other advantage of working with Drosophila neuromuscular junction is that if you look carefully in these structures, you will see that this is a bilaterally symmetrical organism, and you have very stereotyped muscles. So if you look at this structure, and there are certain numbers here, so we identify these muscles, and it's very easy for us to again come back and look at the same structure. Like, for example, in this case, you have muscle 7-6, you have muscle 4. So these have very characteristic innervation pattern, and this is going to be similar across across all these segments. So you can always come back and look at this particular segment in another animal as well. Or like if you want to follow this over a course of development, you can look at the same region again and again, and that will give you a lot of details. So this was kind of an introduction to what the model system is. And again, this is like, if you go further in to one of those rounded structures, again, what is shown here is the active zone in green. So that's the presynapse, and the postsynaptic receptors, or the inotropic glutamate receptors are shown here in red. And you can see that though this is basically a zoomed in confocal image, you can't really make out the pre and the post, but then you see a blurrish region here, and that is where the synaptic cleft is. You're talking of those 20 nanometers. To the right, again, is the electron transmission electron micrograph. In the green labeled structure is the muscle, is the subsynaptic reticulum. So that's like, it provides a cushion for the innervating neurons. And if you zoom into this structure, you see those synaptic vesicles, which I talked to you previously. And the active zone scaffold, that's the Brook pillar that I was talking to you about, that you can see here in green, is actually this electron dense structure, which looks like a T-bar. So just because it resembles a T-bar, it's more often referred to as a T-bar in flies. And then you see this double membrane structure, a thin gap. So this is the 20 nanometer synaptic cleft. And below this, again, of course, you have the muscle and the subsynaptic reticulum. So this is actually, if you go deeper into the structure, this is how it actually looks like. All right. So again, yeah, so this is again a cartoon representation of what I was showing you here. So chemical synapses, as I've already mentioned, enable communication among neurons or between neurons and the targets. And in this case, I'm mostly going to talk to you about neurons and muscles. So it's a neuromuscular junction. And again, just to simplify things, I'm showing you a presynaptic compartment, a postsynaptic compartment. There are going to be proteins across transsynaptic molecules that basically put together this compartment. And again, this 20 nanometer cleft that I'm stressing so much upon. And the reason I'm stressing is something that will be obvious in the next slides. Now, over the course of decades and over the last 40, 50 years, or even more, actually, we've learned a lot about the presynaptic machinery. We've learned about the synaptic vesicle fusion and recycling, and what really happens at the synaptic cleft and the receptors. And most of these information and knowledge that we have presently comes from electron microscopy studies. And this is the conventional electron micrograph, wherein you see the T-bar structure, and then you have those synaptic vesicles. And with advancement in techniques, when we moved on to freeze fracture techniques and electron microscopy, we further understood that this is not actually a static T-bar. Rather, it's an electron dense structure that has got like some spiked appearance. And so you've got different spikes in here, and then you have those vesicles coming and fusing. So all these advancements that have been possible in our understanding of how the synaptic vesicle fusion or the endocytic machinery actually looks like comes from electron microscopy studies. Now, the question is, if you want to really look at something happening in an organism live, you cannot resort to electron microscopy. For that matter, the only option that you have is light microscopy. So there have been tremendous advances in light microscopy, and due to that, what we've been able to really see is we've moved on from confocal, SIMS, TED, and we've moved on to Palm and Storm. So from 200 nanometers, we've narrowed down the resolution to 20 to 50 nanometers, which is remarkable achievement in the field. And just to make you appreciate what this actually looks like. So here is a comparison between a confocal image and a STED image. Again, this is the same structure that I showed you previously. You have a confocal image on the left. Again, it is labeled with it's labeled with two different antibodies recognizing two epitopes of the same protein, that's Brook-Pillard. So you have this protein, which I showed you in the electron micrograph, that looks like a spiked structure. So there are two antibodies here. The one antibody is raised against the C terminus, the other antibody is raised against the N terminus. So you can basically see two different parts of the same protein. And then there are other proteins here, like a rim binding protein. And then you've got a calcium channel, which is called cacophony. And then you've got these postsynaptic receptors. So you've got these different structures across they are basically placed in an axial orientation. And using super resolution microscopy like STED, what people have been able to show is the distance between these different components. Like if you look at the distance between the postsynaptic receptors to the extreme end of Brook-Pillard, you'll be able to see that this is somewhere between 170 nanometers to 170 to 180 nanometers. And then individually you can make other calculations and see how far each particular protein is. So that is remarkable. That's really good. But then the issue here is that with the advancement in high-end microscopy, we have not been really able to keep pace with our staining procedures. Now, the microscope that have been developed and the amount of advancement that is going on is remarkable. But then if you're not able to keep pace with our labeling strategies, then this whole advancement is going to be basically a waste. Now, what do I actually mean by that? So what I'm trying to tell you is here, the normal conventional immunohistochemistry that you do, you have a protein of interest, you've got different epitopes, then you use a primary antibody. So this Y-shaped structure is the primary antibody. And then to recognize this primary antibody, you add a secondary antibody, which is conjugated to a fluorophore. See, overall, the distance here is like 30 nanometers. That is primary plus secondary itself is 30 nanometers. Okay. And then you have these different compartments. And again, now I think you'll be able to appreciate why I was stressing so much on this 20 nanometers. So you have this 20 nanometer synaptic cleft. And then say you are studying a presynaptic protein, you are using this conventional staining procedure. Now you have the epitope, of course, binding to this place. This primary secondary coming, and then you have the fluorophore, which is at least 30 nanometers apart. Now, when it is 30 nanometers apart, already away from the original epitope, and when you do a confocal image, what you're going to see is you're going to see a blurry kind of a thing. And again, that 30 nanometer distance itself is bigger than this 20 nanometer distance that we want to study. So there itself is the problem. So how do we get across that? So one solution to this problem is having something instead of that can bypass this 30 nanometer problem, and we have something much smaller. So that is called as a nanobody, and these nanobodies are highly specific, and they'll bind to particular nanotag. So these nanotags need to be like incorporated in your protein of interest. And then, of course, you can see that instead of 30 nanometers, now you're narrowing it down to just 2 to 4 nanometers, which is remarkable. Now, when you want to study something which is like across a distance of 20 nanometers, it's going to be far more easier and far more reliable. And it will help you tell whether your protein that you are studying is a presynaptic protein or a postsynaptic protein. So now, what are these nanobodies? These nanobodies are variable chain heavy only fragments. So you have a traditional Y-shaped immunoglobulin structure, but these nanobodies are just the heavy chain fragment. And these are produced in the camelid species, mostly camels and alpacas. So apart from the traditional immunoglobulins, they also produce these heavy chain only antibodies. And that's why, they offer a wonderful resource for high-end microscopy. And so with this kind of an introduction, I'll now step on to what I'm going to talk to you mostly about, and that is this new tag and its corresponding nanobody that we call as the ALFA-tag alpha-nanobody pair. And some of the results that I'm going to show you is we've used this method to tag a membrane protein called neurexin-1. If you remember again, in one of the initial diagrams, I showed you it's a transmembrane domain protein that binds to neuroligin on the postsynaptic side and bridges that to 20 nanometer distance. And we made some of these transgenes, and we compared between the two differently tagged neurexin variants. Like we had neurexin alpha tag, neurexin GFP tag, and we made a comparison to see how good the other one is. We also performed function analysis, and we also did an endogenous tagging using CRISPR rather than the overexpression studies. And we did a very cool experiment where we showed that we can also, using this technology, do an in vivo reconstitution of a full-length protein. And that was, I think, the coolest part of this whole study. And then I'll, of course, end with a little bit of future directions. Okay. So again, going back to the structure that we were really interested in studying. So when I started my postdoctoral work, I was interested in studying this particular protein called Netto. Now, this is auxiliary subunit protein for the inotropic glutamate receptors. And the predominant form at the neuromuscular junction is the Netto beta. And this is, again, as I said, is a postsynaptic protein. Again, it is a membrane, it's a transmembrane protein. And the biggest problem with transmembrane proteins is that it is difficult to tag these proteins. And we tried tagging this protein with different fluorescent tags like GFP, RFP, but then somehow the tag would fall off or it get cleaved. It would never really work. And even if we were able to get it, get it to be produced, what happened was somehow it would not work. That is, it would not be functional. So we were really looking for a strategy that would help us to tag this particular protein and overall tag most of these difficult proteins. Okay, so yeah, we were reaching out to different companies and different resources, and what we came across was one particular company in Germany. So that's Nanotag Biotechnologies, headed by Dr. Felipe Opazzo and Dr. Stephen Frey. And they had developed this particular ALFA-tag alpha-nanobody pair, and they were doing quite a lot of experiments on cell culture model, and they had very promising results. So we reached out to them, entered a collaboration, and we said, we'll try this in flies, we'll try this in Drosophila and see whether we can do something in vivo. And what I'm showing you in this table are the commonly used epitope tags. You've got the flag, HA, MIC, polyhis, GFP is the most commonly used, and then other spot tag and EPA tags that are available. So they are like, they come in different sizes. There are different properties, like whether it's being resistant to fixation and the specificity of the antibody and the nanobody availability. Of course, you have a nanobody for GFP available now, but then that also has other problems. And then, of course, so today I'm going to talk to you more about this alpha tag pair, which is, again, as you can see, it's a 15 residue long particular tag. Okay, so now how did they come across this alpha tag sequence? So they had a very stringent criteria that it should be a small monomeric and highly soluble electroneutral protein. It should be resistant to fixation. It should be well expressed, and most importantly, it should be unique. So this kind of a sequence should not be available in a eukaryotic or prokaryotic host, so that with that you make sure that there is no non-specific signal that you get. And then the beauty of this tag is also that you can tag your protein at the N terminal, C terminal, somewhere in between, anywhere, and that's a big advantage. And then you've got these different prolines on either side, so that they basically shield this tag from interactions with the neighboring residues, and it has a compact alpha helical structure as well. So, and then of course you follow the same procedure of immunization of the alpacas, and then you do a selection screening, you isolate the B cells, and then you run them over these alpha-tag beads. You have these B cells bound to beads, you generate a library, and then you screen those library, and then you screen for the corresponding antibody or the nanobody against the alpha-tag. So you have the alpha-tag sequence, you now also have a corresponding nanobody that specifically binds to or recognizes the alpha tag. And then after this whole process, they also had the sequence of the nanobody, which is around 120 to 130 residues big. And these are some of the experiments. You can always go back to the paper of Gosge et al., Nature Com 2019, and you'll have a lot more explanation for the thing that I'm going to talk to you about. So you've got these alpha-tag proteins, and they retain their normal subcellular distribution in tissue culture experiments. So this is again COS7 cells, and you've transfected these cells with the TOM70 EGFP and alpha. So this is outer mitochondrial membrane protein, and you've got this transmembrane domain within the mitochondria. You've got this EGFP and alpha-tag together, and they have imaged both this EGFP signal and the alpha-tag signal together. And so since this is the same protein and it is tagged by two different probes, you should see a very good level of co-localization between the EGFP and the alpha-tag, and this is exactly what you observe here. Now, again, they've also done a similar kind of experiment, and this is, of course, a super resolution microscopy instead. They have labeled, or rather they have expressed a flag-tagged alpha-tag vimentin. Now, vimentin is an intermediate filament protein, and you see those wonderful, fine structures that is usually associated with vimentin. And again, so this also shows that this particular tag and this particular system is amenable also to super resolution microscopy. So it's not only specific, it's also good for super resolution microscopy. Now we're talking of affinity of these nanobody alpha. So again, so this is a mannose binding protein, this expressed in E. coli. And what they've done here is they have attached all the different commercially available probes like HA, FLAG, MYC, and alpha. And they express this particular protein in E. coli, and then they did a spot assay. And here what you can see is they have used an anti-MYC, anti-FLAG, and anti-HA antibodies, and then further they came with a secondary antibody to detect this particular protein. So as you can see, there are two steps. You do a primary and then you do a secondary. So after two steps, you can look at how the signal actually looks like. But with nanobody alpha, you don't need a second amplification step. You have an alpha expressing protein, you directly put the secondary as nanobody alpha, which could be attached to a particular fluorophore, and then have a look at the signal. So it's actually some tenfold better signal that you get with the nanobody alpha. And again, this is a quantification of the same, and you see that it is highly specific, and it is a very high affinity for the alpha tag. Now, how does this interaction actually take place? So you've got this particular peptide, which is the alpha peptide, and then you've got this nanobody, and it is recognizing this alpha-tagged peptide using its complementary determining regions, CDR3 and CDR2. It actually, you've got these 15 residues. These 15 residues, all these 15 residues actually interact with this CDR2 and 3 regions of this nanobody, and that results in a very tight interaction, and you have a very high affinity for the resulting interaction. So I've talked to you about a little bit of the biochemical and structural features of the tag and the alpha nanobody, and now I'll move on to the results and what we did with the neurexin protein and how did we detect it. So neurexin, as I've already mentioned previously, is a key synapse organizing molecule. This is a Drosophila neurexin. It has got this lamin G binding domain and the EGF binding domain. You've got a transmembrane domain and a PDZ binding domain. This is the human neurexin. And even the human neurexin has a similar domain organization, and these numbers actually tell you about the percentage identity. So these particular domains across humans and flies are 32% identical. So overall, the structure is the same. They have got very conserved extracellular domains. You've got transmembrane domain, and then you've got the intracellular domain as well. And again, as I've said, these are huge extracellular proteins. So these are very difficult to tag, because if you insert a tag in any of these regions, you're most likely going to alter the function of that particular protein, or probably even hinder the interaction that it has with other proteins. So the best place to tag these proteins could be the small intracellular tails of this particular structure. So and of course you have the small PDZ-binding domain in neurexin, which is again very highly conserved. So that actually makes it even more difficult, because you cannot really touch this extracellular part of the protein. The intracellular part, there are certain conserved structures, and again you cannot play around with this structure. So you're left with a very small region where you can probably put your tag. So that is kind of a issue, and it makes it really challenging. So we did a phylogenetic analysis of this Drosophila neurexin, and what we found, again, this is only the intracellular part that is being shown, and we in here which has a stretch of glycines, and we thought this is probably the best region to actually insert this tag without hindering its function. So we did a secondary structure prediction. We ran algorithm actually, and when we put this alpha tag here, what we found was that it introduces an alpha helix as was predicted, and it does not alter any other structure. So the sheets and the turns are kind of conserved, and you just introduce a small alpha helix in the structure. So that kind of gave us the confidence that I think everything is going to work fine. So we went ahead and made particular transgenes. So we had this neurexin 1 GFP already available, previously published. We made two different transgenes. One was neurexin 1 alpha, that is with this alpha tag at the C-terminal domain, and one we had a neurexin 1 that did not have any particular tag. And the strategy that we followed was to test the functionality of these transgenes in rescue experiments. So you have a mutant organism that does not have neurexin, and you insert these three different transgenes and see which of these actually work, which of these makes the protein functional. And then of course we evaluated the localization of these variants, and we also saw tested how these different tags could be applicable for super resolution and live imaging. So so if you look at the neuromuscular junction of a Drosophila neurexin mutant, this is a wild type, this is a mutant, you can appreciate that this is much smaller. This entire structure itself is much smaller compared to the wild type, which is kind of longer. And so we repeated this experiment in our hands, and we found a similar result. This is the control. You have a longer axonal arbor, whereas in the mutant, you have this much smaller. So now the next experiment that we did was we tried to rescue this mutant using these three different transgenes that we have mentioned. Like one was having a GFP tagged neurexin, one was having the alpha tag, and the other one was not having any tag. So if you look at the overall morphology, all the three transgenes equally rescued the morphological phenotypes. Again, this is quantified here. You look at the Bouton number, that is this number of this round varicosities, and the length of this entire NMJ. So in this case, in both these cases, you see that the three different transgenes were equally effective in rescuing the phenotype. Now the question was, that's good, it works, it rescues the morphology, but what about the function? Does it also rescue the function? So for that, we resorted to a technique called electrophysiology, wherein we try to look at the potential from the muscles in these particular animals. And what we then observed was, this is the control, and this is the mutant. Here you see that this spike is much smaller. So the evoked potential, this is one of the parameters that we look at. So evoked potential is much smaller in the mutant. And when we put the three different transgenes, we see that the untagged version and the alpha tagged version give back value which is pretty much similar to the control. But whereas this GFP tag transgene, now if you remember in the previous slide, I showed you that this rescues the morphology. So now we were trying to see that whether it also rescues the function, but what we observed was that the GFP tag transgene was not effective in rescuing the function. So this pretty much resembled like the mutant. Again, this is quantified here. You see the red bar and the green bar, they look pretty much similar. So there's not a significant amount of rescue that is observed. Whereas the magenta and the blue one, so these are the untagged versus the alpha tagged, you see that there is a substantial amount of rescue. Now that tells us that, when you have an untagged and an alpha tagged version, it also is, the protein is also functional. But if you look at the other proteins, if you look at the other proteins, I mean, if you look at the GFP-tag protein, that somehow does not rescue the function. Now, of course, we were interested in knowing why does that happen? What is the reason behind this GFP-tag version protein not being able to rescue the function? So we resorted to super resolution imaging. Again, these are STED images that we captured using like a SP8. And you see here, in the green, this is a signal for the alpha tag. And in magenta is the signal for the neurexin protein itself. So we used the nanobody alpha to detect the alpha tag, and that of course that also detected the neurexin. And we used a traditional neurexin antibody to also see the neurexin pattern. So this is how it looks like when you overexpress alpha neurexin tagged with alpha tag. And this is how it looks like. You see the very distinct punctate patterns. Whereas if you look at the GFP tagged neurexin, the first thing that you will see here is that this punctate structure is something that you don't see. You see it a lot more diffused. And also, what I'll show you in the quantification is that the level of co-localization is going to be pretty much less compared to what you will see here. Now, yeah, so this is where the quantification is. So you see the co-localization between the green and the magenta channels, which is like 93%. It's really high. And if you see the co-localization here between GFP and neurexin, it is 54% approximately. So this is really less. Now, how is this possible? So this probably what we think what is actually happening is that you have a bigger tag like GFP, and somehow this GFP is falls off this particular protein. And once it falls off, it appears very diffused. And also because it falls off, it is not able to rescue the function. So that could be a possible explanation why the GFP tag version is not able to rescue the function. So of course, now we know that, okay, using this alpha tag, we can also use to visualize this particular protein using super resolution microscopy. That's a big plus. Now, moving forward, what we then wanted to do was to do endogenously tag neurexin alpha and function analysis of that, because these are again overexpression experiments. They tell you that the system is working, but again, overexpression is not a very clear representation of what is happening inside a cell. So for that, what we did was we performed endogenous genome editing using CRISPR. And what you'll appreciate here is this is the control. So it doesn't have the edited neurexin. So the only signal that you see here is using the neurexin-1 antibody. And this is the ventral nerve cord. And you don't have any signal whatsoever for the nanobody alpha. And the endogenously edited tagged protein, what you see here is, again, this is the neurexin. This is endogenously edited, just stained with the alpha tag, alpha nanobody. And this is the neurexin protein itself. And then you see a very high degree of co-localization between the magenta and this gray here. And that's how it looks like. And we also generated in the process a truncated version that did not have this PDZ-binding domain. Surprisingly, what we found in this mutant is that you have those, you still can see the nanobody alpha staining, but then these are confined to particular compartments. You don't see any staining in the neurexin itself, because that's a C-terminal protein, and this epitope lies somewhere here. So it is definitely the PDZ is not there, so it's not able to bind. So you don't really get a signal out here. But what is really striking is you see here that there are these round structures which we think is the endoplasmic reticulum. So when you don't have the PDZ binding domain in neurexin, it ends up that these protein probably gets stuck in the endoplasmic reticulum, and it is not able to pass the quality control of the cellular machinery. So it basically gets stuck, and that also results in functional defects. Now, moving ahead, as I was talking to you about one of the coolest part of these study was we then thought that the nanobody itself has a very strong affinity for the alpha tag. So what if we just have this PDZ binding domain and we have the nanobody alpha? So this is a chimera that we made, nanobody alpha with the PDZ. And we provided this thing, this chimera in the neurons. And we asked whether just providing this chimera itself could restore the functionality. That is, if you provide only the PDZ binding domain using a nanobody alpha, whether this can actually bind to this truncated neurexin and restore the full function of it. And that is what we did. It was a bold experiment, and what we found out was that actually when you do this experiment in this neurexin 1 delta PDZ background, when you supply or when you provide this nanobody alpha PDZ domain in neurons, it is actually able to reconstitute the full length neurexin. Now, this is remarkable because the staining here looks exactly identical to the neurexin 1 alpha endogenously tagged allele. And you can also see that the neurexin pattern in the mutant, you see that there is no such signal, but in the neurexin C-terminal staining here, you can see that the staining is almost back. And well, it's not as good as what you would see in the control animals. That is because it's overexpression system, and a lot depends on the level of expression as well. But the fact that you can see this particular staining in the dendrite field also tells you that this particular antibody that binds to this epitope tag, now if you don't have the remaining part, it will not be able to bind, like in this case. But here, since you have actually reconstituted a full length protein, your antibody is able to bind, and that's why you're getting the signal. So this was I think one of the remarkable features of the study. So we can basically reconstitute in vivo. You can have a full length protein now. Now, what we also wanted to do was look at the neuromuscular junction. And again, what I'm showing you here is the control that does not have any tag. The neurexin 1 alpha, again, we are able to detect it at the nerve terminals as well. And in the delta PDZ version, you're not able to see any signal. But again, when you provide it back using the same method that I talked to you about earlier, you can see that the nanobody alpha signal is back in the neurons. And again, most importantly, this is a single step detection, which is again very important. You're not using a primary, secondary fluorophore tagged method. You're just using one antibody, and then indirectly you are able to visualize this. So this is remarkable. Now, the other question was also like, is this just the morphology or is it also the function? So we did electrophysiology experiment on this also. And what we did find was that in the rescue animals, we were able to get back the values almost related to the control. So that showed that, you know, not just the morphological features, but also the function can be restored when you can reconstitute the full length protein. So this has been the cool part about the study. Now, yeah, just to sum up what I have shown you so far is that when you insert this alpha tag, it preserves the normal distribution and function of the synaptic proteins. Importantly, you can also detect this protein in one step. That is again very important. And this is important for low abundant proteins. And again, this important characteristic would be that you are able to reconstitute the full protein and able to restore the proper synaptic distribution as well as function. And using this method of reconstituting the full length protein and similar kind of studies, what we can do is we can study multiple structure function applications. Like very briefly, if I have to tell you, like you can do biochemical experiments where you can truncate different parts of the protein and selectively bring certain parts and see which part of the particular protein is important for which particular function. So this is like classic biochemistry experiments that you can actually do. Now, moving ahead, what we also wanted to then look at was, can we express this nanobody alpha itself within a cell in vivo and then see what really happens? So now this is where instead of providing an antibody from outside, we are trying to make a system wherein the antibody binding happens inside the cell itself. Now, so how does this work? Again, this is cell culture studies from the same group. And what they have shown is they have transfected these COS7 cells with the TOM70 EGFP alpha and the nanobody alpha scarlet. Now, this alpha is supposed to bind to this nanobody alpha is going to supposed to bind to this alpha tag. And so if the binding is good enough, you're supposed to see the red and the green signals perfectly co-localized with each other. And this same study was also done on vimentin. As you can see here, the two signals, and then this is the merged image. There is a perfect co-localization between the two signals. That tells you that, this nanobody scarlet is able to follow the Tom70 EGFP or the alpha vimentin in this case and able to bind to it. So this is again another remarkable feature of this tag. So we then again wanted to test this in the flies. What we then did was, again, this is a control setup. This is the ventral nerve cord. As a control, what we did was we had a neurexin, which was not tagged with anything. And then we had the nanobody scarlet. So obviously, there is no binding partner for the nanobody, so it is not going to bind to anything. As a result, what you see here is the red signal is going to be diffused all around in the cytoplasm. And if you see a colocalization between the neurexin and the scarlet signal, it's hardly 6%. Whereas on the right, you have the neurexin-1 alpha tag protein, and you are also providing the nanobody alpha M scarlet. Now, this alpha scarlet chimera can bind to this particular protein, as a result of which you see that now the signal is not diffused, but it's rather concentrated along the dendrite field. And again, if you look at the co-localization from 6%, it has moved on to almost 63%. So this shows that the system actually works. Now, what actually happens in the nerve terminals? Again, a similar set of experiments. Control, no tagged with the nanobody alpha scarlet. You see that the signal of scarlet within the synaptic bouton is diffused in the cytosol. It's all around. And you have just 7% co-localization between neurexin and the scarlet signal. Whereas on the right, what you see is a very distinct punctate distribution of M scarlet nanobody alpha. So, and then when you do a further merged image, when you see that with the neurexin antibody, you see that this has gone to 66%. So this shows that when you have alpha-tag protein and you provide a nanobody with a particular chimera, now this is going to follow this alpha-tag protein and bind to it very specifically, as a result of which you get this punctate distribution. So far so good. This shows that you can actually make it work like an intrabody, wherein if you express the alpha-tag protein and also if you express the nanobody within the same cell, it's going to bind very specifically within the cell, and it can work as an intrabody. Now, we also did some live imaging, because the whole purpose of trying to use an intrabody is to do some developmental studies and do it over a period of time. So again, so these are two videos that I'm showing. This is a ventral nerve cord. You can, the entire length, you see the wire-like structure is actually the axon. And I'm just going to play the video. So this is the experimental animal. Again, you have the alpha-tag protein. You also have the nanobody with the scarlet. So basically, the hypothesis is that this is supposed to bind to this alpha-tag protein, and you are supposed to visualize it. Whereas in the control animal, you don't have any. So the nanobody alpha is just going to be diffused along the cytoplasm. So now I'm going to play the video, and what you can see is you can see these two vesicular structures that are moving along. So this is the nanobody alpha scarlet bound to the neurexin alpha. So essentially, this is neurexin that is moving along the axon, whereas in the control animals, you don't see anything. So what you see is just going to be some background scarlet fluorescence that you see. And also, you look at the chymographs here, you can see a nice chymograph that shows the movement of the particles, whereas in the control animals, you don't have any such movement. So this is how it actually looks like. So next, we wanted to really ask, like, what is the nature of these vesicles? So there could be like multiple autophagosome-like vesicles, or it could be, early endosomal vesicles, or it could be other vesicles as well. So we wanted to study the nature of these vesicles, and what we then did was we did a double labeling, wherein we also performed experiments with the say RAB7 EGFP. So what I'm showing here is something with RAB7 EGFP, and This is the scarlet fluorescence, this is the RAP7 fluorescence, and this is the merge. So you can see that the two signals are pretty much separated out. So you have some signal coming from the scarlet. Again, you have these vesicles moving in, but they never really co-localize with the green signal coming from RAP7. So we tested quite a few of these molecules. We tested RAP7, RAB11, ATG8, cholera toxin, and in none of these cases we could find any kind of co-localization. So this is still something under study. We still do not know the nature of these vesicles, but in the near future, hopefully we'll be able to really find out. Okay. So to conclude, what I've really shown you today is that this alpha-tag nanobody alpha pair is a versatile tool for tagging and detection of synaptic proteins, both in vitro and in vivo. And it has got a very high specificity and affinity for the alpha epitope. We did our experiments on neurexin. We did an overexpression, as well as we did endogenous tagging experiments. And in both cases, we were able to do this in vivo single step detection using both confocal and super resolution imaging. And what we learned from this study was that the deletion of the PDZ-binding domain itself disrupted the localization of the neurexin 1. And when we expressed this PDZ nanobody alpha chimera, we were able to restore the proper localization and function, telling us that we could use this technology or this method for in vivo reconstitution of synaptic proteins. And what we also did was we did some live imaging experiments wherein we have shown that this can function as an intrabody. That is, it still retains its binding ability to its epitope in the cytoplasm. And we use this method to do some of the live imaging studies, which I've already shown you. So moving forward, in the initial phases or in the initial slides, I told you that I was trying to understand, or rather tag, this particular protein, which is a postsynaptic protein called NETO. And since we were not able to really tag that, we were looking for methods that would be helpful for us. And in the process, we ended up using this alpha tag, and we used neurexin as a as an experimental protein wherein we could just see whether things work out or not. And neurexin, being a transmembrane protein itself, was a difficult protein to tag. And since we were able to do that successfully, we thought of trying it out on the protein that we were initially interested in, that is neto beta. And what I'm showing you here is again a neuromuscular junction image. So these are SIM images captured on ELIRA 7. And what you see here is when we had this transgene, there is a nano beta tagged with this alpha tag, and we expressed it in the muscles. And we then labeled it using this nanobody. And you see that these are wonderful receptor organization patterns that you actually see at the postsynapse. So if you were to do a receptor staining, you will get a similar kind of a staining pattern. And this is the presynaptic protein Brook pillar, which I showed you many a times. And you can very nicely see that the pre and the post is kind of separated along this with a 20 nanometers kind of a distance. So again, coming back to the problem that we started with was this 20 nanometer issue and how to resolve it. So and this is not even this is not even like STED. This is of course super resolution, but this is a SIM, and even in SIM you're able to get a good quality image. And so if you move on to super resolution, the distance between the pre and post would be even more better. So this would be really helpful. This technology would be really helpful, not only for us, but other people working in different fields. And this is definitely not restricted to only Drosophila. So they're going to use this for doing a lot of structure function analysis, and also understanding how these receptors and other PSD components are actually assembled and stabilized at the synapses. So this is like one of the future applications that I've kind of shown here, but overall there would be many applications, and one of that of course would be the live imaging experiments that I showed you. So with this, I would acknowledge my mentor, Dr. Mihaela Serpe, in whose lab this entire work was carried out. And I'd like to particularly mention and acknowledge Rosario Vici-Domini, most of the live cell experiments and the microscopy work that we did, we were doing it together. And he is a wonderful cell biologist, and he's been very actively involved in, upgrading methods and making new, trying to come out with new technologies for visualizing synaptic proteins. The functional analysis, wherein I talked to you about the electrophysiology, were performed by Tahi Han. He's a staff scientist in the laboratory, and he's an electrophysiologist. So without his help and without Rosario's help, these studies would not have been possible. And of course, most importantly, I'd like to thank my mentor, Ella Serpe, for all her support and encouragement and her guidance throughout this project, and all the other Serpe laboratory members. And particular thanks to Vincent Schramm, who's at the NICHD core microscopy facility, who's helped us with the live imaging. Again, those images were captured with the LSM 780 Zeiss. Again, Daniela Malide, who's at the NHLBI microimaging core facility. So she helped us with the STED super resolution imaging. And this work, of course, is a collaboration with Dr. Felipe Opazo at the University of Göttingen and Nanotech Biotechnologies. So with this, I would end my presentation, and thank you very much for this wonderful opportunity.
Thank you, Dr. Saumitra. I think this is a very extensive presentation which I've come across after a long time, and a very big congratulations to you for this extensive work. I think it would definitely mean a lot to the upcoming budding researchers who are looking forward to work in the neurology field, especially with a special emphasis to how imaging can be an integral part onto resolving a synaptic difference. So the synaptic gap which you have shown as a 20 nanometer, and how you have overcome that difference with nanobodies, I think that is very fascinating.
Yeah, it's pretty challenging. Like, I mean, you can't really get it to work immediately. You have to do a lot of, standardization of the experiments. And I mean, you don't really know. In the cell culture, it looks good. It looks like it is working. But in living systems and in vivo, when you want to do something, it's really tricky. And there are a lot of issues, like, a simple issue could be like where you're putting the tag. That itself might be a problem. So yeah, I mean, it's pretty much, it's tricky, and then it's also very challenging. So, and then of course, once that is working, going to the microscope and capturing those images and setting the right parameters, of course, so I mean, I think this is going to be a wonderful tool for cell biology work, and I think a lot of other people also would be using this technology. Yeah.
Okay, but before we end this session, can I have like a few questions? So can I?
Yes.
Okay. So like it may be very like a naive question, but how do we stain acetylcholine esterase or like choline acetyl transferase in a neuromuscular junction, for example, in case of a Drosophila?
So so what you've talked about these are the enzymes, right?
Right. So if you are looking to staining enzyme, how do we do it, or what fluorophores would be very ideal for that?
So for the enzymes, I'm not sure if you can directly do an antibody kind of a thing, because enzymes are going to be like, they just do the work and then they are done. So they are not going to stay in a locked kind of a situation for like what you have with other proteins, which may be like on the membrane. They are just going to come do the work, and they are going to basically dissociate out. So one way of probably looking at it is, if you know what are the end products of the reaction, so you can have particular proteins or particular sensors maybe, and you can it will still not be a direct method. It would be like a proxy, that you are looking at the end product and trying to tell that, okay, the enzyme must be there because this product is formed. So maybe that is one way of looking at it. And the other, if you again if you are doing some live kind of studies, and if you have say particular substrates, for example, and if there are certain substrates which are like in a locked kind of conformation which do not fluoresce, and they would only fluoresce when they are acted upon, like there is a particular substrate and you have a fluorophore in that substrate, and that substrate is acted upon by some enzyme, that particular enzyme that you are interested in, and when the enzyme acts on it, say there is some chemical bond that breaks or something of like that happens, and the fluorophore is exposed, and you can then see the signal, and then you say that okay, now the enzyme is there and that's why I can see the signal. So that those kind of things are possible, but if you're asking me the exact compound or the exact thing, well, I have to look that up. Yeah.
Okay. And how easy or how efficient it is to express nanobodies? What is your experience?
So the nanobodies, if you know the sequence of the nanobody, it's not a big deal. You just have to clone it, and then you have to just, if you want to make a transgenic fly, I mean, you just clone it, you put it in a particular vector, and then you send it to the company. They will they will inject that plasmid into the flies, and then of course you will have that. So knowing the sequence is important. So that is one thing. The second thing would be like how efficiently that is going to be expressed. That is another question, because if the expression is not good, then it is not going to be helpful. Right. So for that, you don't really have much that you can actually do about it. You can probably when you are making this clone, what you can try to see, you can do a lot of codon optimization and stuff. Make sure that the right codons or the codons that are extensively used, they are the ones that you put in your particular sequence. And when you want to make transgenic animals, when you want to insert that particular, say, plasmid in a particular region, so you need to know that that region should not be, in a silenced region or something, like it is not accessible, it is under a chromatin locked state. So then that would be another problem. So these are like, there are resources available. You can study that, and you can try to find a particular location where you want your the plasmid to actually be docked. So those resources are available. So you can try your best to make sure of on your part, take all necessary precautions to make sure that the expression is good enough. But then again, you will only come to know about it when you do the experiment, whether the expression is good or not. So,
right.
Yeah.
Okay. Thank you for your time. Thank you for this wonderful presentation. If you have any questions, like especially I'm addressing to the viewers. So if you have any questions, please write it in the comment section. We'll definitely get back to you. And if you're looking for any suggestions to your work, you can also feel free to you can mail me, or you can write in the comment sections also. I'll definitely get back to you. So with this, thank you, Dr. Saumitra, for your time and for a beautiful presentation. And with this, I also would like to extend my sincere gratitude to you, so that you have agreed to come to this platform on a very short notice. And I'm really thankful to you. Looking forward to have more sessions like this.
Thank you. Thank you.
Thank you, sir. Thank you.